Hi, everyone, and thank you very much, Ahmed, for agreeing to give a Build a Cell seminar. And you can get started.
It's my pleasure. Thank you for the invitation. And it's my first time here. So Kate was telling me earlier there's a Q&A session afterwards where we can discuss a lot of this stuff. So this is meant more to introduce, hopefully, many people to some of the work that we do and then lay a foundation for a lot of questions later on. My lab has been at Scripps for a few years now, and we're interested in a number of different topics. Today, I'll tell you a little bit about our work to create systems that really allow us to programmably biosynthesize what I'll very broadly call new to nature molecules. Hopefully, by the end of the presentation, I'll have convinced you that that is actually the case. But you'll see as we progress through that this has really been a labor of love where we're trying to build out the components at each step and try to understand exactly how to maximize their function. A lot of our work in this space comes from a desire to make macrocycles. Of course, recently in particular, there's been a resurgence in trying to pursue peptides, not only in academic science, but also in the pharmaceutical space. And historically, some of the greatest successes from macrocycles, sort of not counting the modern era, have come from natural products that are often made by these massive complexes like NRPSs or non-ribosomal peptide synthesis. And historically, these machines have garnered a lot of attention because there's a code to how they select their building blocks and how they sort of polymerize them and build really interesting natural products. But it actually turns out to be incredibly difficult to re-engineer these complexes to make new to nature small molecules like macrocycles. And so as a result, researchers have developed a number of more contemporary and sort of compatible strategies that allow us to access increasingly complex natural products like molecules, but they're almost always derived from natural translation. These make use of some interesting post-translational modifications or clever chemistries to constrain the way that these peptides are built. So one of the most sort of quintessential classes in this space is RIPS, or ribosomally synthesized and post-translationally modified polypeptides. These are made by the ribosome. They're often functionalized in some way after that. And in many cases, you can see here in this niacin structure, they can result in really interesting microcyclic structures that are important for their natural activity. There are increasingly synthetic strategies to do this in Cyclops and in phage display, where post-translational modifications can allow researchers to access these constrained cyclic more topologies that actually do end up being really critical for function. But by and large, these systems suffer from a specific issue, which is that they are almost always built from the natural 20 amino acids. And even though there is significant sort of post-translational modifications to these peptides, they are constrained in the chemistry that they can provide access to. Now, fortunately, these technologies have been supplemented with yet another long-standing area of research, which is called genetic code expansion. And for those of you that aren't familiar with this space, this is the fundamental premise that you can design components within a living cell, or sometimes even in vitro, that allow researchers to incorporate new to nature building blocks during protein translation. Historically, these have been non-canonical amino acids, but increasingly they're sort of going into the space of what are generally called non-canonical monomers. These are ones where the backbone itself is also modified beyond the side chain. To access this technology, you need to satisfy a number of criteria, but the most important of which is the creation or the use of what are called orthogonal tRNA synthetase pairs. The synthetase is what selects the amino acid, the tRNA is what is used by the ribosome to do translation, and the end result is this incorporation of this hexagon that you see here, which supplements the genetic code with new amino acids. And the way that these are developed is by this now classical cascade of positive and negative selection stringencies that allow you to redesign the active site of the synthetase to use this new to nature amino acid. And this strategy has been wildly successful. It's resulted in hundreds of different non-canonical amino acids that have been incorporated into proteins. And this has led to a number of innovations that allowed us to study not only natural biological systems, but also innovate beyond them to do some interesting chemistry. And so on the one hand, these non-canonical amino acids have allowed us to understand how natural systems adapt to agents that might exist in the cell, and how these agents can modify natural amino acids like you see in this image. They've allowed researchers to overcome natural bottlenecks in developing, in this case, TNF-alpha analogs that incorporate non-canonical amino acids and have slightly altered responses in living animals. They've also allowed researchers to really try to understand some of the more nuanced relationships that exist in signaling networks inside of living cells, especially as a result of post-translational modifications. And perhaps the poster child of this is the histone modification, the histone code. And so this is just a small subset of the types of modifications that can happen. But of course, what genetic code expansion allows us to do is to try to integrate these into histones and try to understand the impact on biology and chromatin. And more recently, a lot of these fundamental innovations have allowed researchers to go the next step and to invent new bioactivities that really extend beyond what nature is capable of. And this is just one more recent example where you can see here, there's a catalyst that does a certain reaction, and the researchers chose to incorporate this N-methylhistidine that allows them to increase the reactivity a little bit, but then on top of that, with directed evolution, they can achieve rates that are considerably higher than the natural catalyst. And so all of this is to say that genetic code expansion really supplements the diversity of building blocks that exist in nature and allows researchers to start to explore new and interesting biology. But all that being said, there are a number of caveats, especially as we start to think about building comprehensively modified new to nature molecules in the lab. Often these are restricted to specific classes of non-canonical amino acids. Those are typically sort of bulky side chain amino acids. The efficiencies are often quite low, and perhaps most importantly for many applications, the multiplexability is quite low. And this has to do with the ability to assign a new building block to a new codon, which I'll talk about on the next slide. There are often also context-dependent issues that remain difficult to deconvolute from the rest of the machinery in the cell that can result in overall efficiency being quite low. And so all of these issues have, I would say, limited the impact of genetic code expansion on not only applications in the protein space, but also creating new to nature molecules altogether. So this codon problem is perhaps the longest standing issue to date. And what I mean by this is if you think about the genetic code, which in genetic code expansion, we're going to modify so that we can assign a new building block to an existing codon. The genetic code comes pre-programmed in every organism. And as a result, what the researcher must do is to reassign a pre-existing codon to a new amino acid. And there are a number of strategies that have been developed to really overcome this bottleneck. The most traditional one of these is what's called stop codon or amber codon suppression. In this case, a tRNA is designed or engineered or evolved to read the amber stop codon. And the reason this stop codon is chosen is because it is often the lowest usage stop codon, the least abundant stop codon in things like E. coli or yeast or human cells. More contemporary strategies are a little bit more complicated than this. So one of the most successful to date has been genome rewriting. Rather than competing with a pre-existing central dogma, researchers can rewrite whole genetic codes to eliminate codons, and in doing so, repurpose them to new building blocks. As you might imagine, of course, this is a heroic undertaking that requires a lot of patience, but also significant overhead to build out complete genomes from scratch. Another really interesting strategy has been the development of these synthetic nucleobases. And so one way to think about this is if the central dogma, which is composed of A, T, C, and G, already exists, adding new codes allows you to now add new codons. And that addresses the issues that you see on the left, where you no longer have to worry about crosstalk with the cell, but rather by adding new genetic information, you can add new amino acids right off the bat. These have additional issues, of course, which the biggest of which is that the researcher must synthesize all of these building blocks and provide them to live cells. And the moment that they're taken away, that information reverts to the natural central dogma, one of the natural nucleobases. And finally, there's this strategy called quadruplet codons, and it sounds exactly like what it's called. Under very weird conditions and often with very poor efficiency, the ribosome can actually go beyond the triplet genetic code to read a 4-base codon. You might imagine as a result that if this were generalized, that rather than having to worry about the 64 triplet codons, quadruplet codons would provide access to 256 unique possibilities, and in doing so broaden the scope of genetic code expansion to new codons at will. So all of these strategies have been successful in their own right, but they also all have unique liabilities. And this table is just really meant to summarize some of the strengths and weaknesses of these different technologies. In our lab, we got interested in quadruplet codons in particular a few years ago, primarily because it has really interesting sort of strengths that make it potentially extremely generalizable and very easy to access. But it probably has the biggest liabilities in predictability and overall decoding efficiency. And so we started to, a few years ago now, tried to look at what makes quadruplet decoding more or less efficient. To contextualize this, I want to tell you a little bit about how this whole field arose. And it's extremely old, and it arises from an observation that actually has nothing to do with genetic code expansion. In a series of studies that were published in the sort of mid-70s to early 80s, researchers were interested in exploring how bacteria in particular, but also later single-celled eukaryotes, adapt to stressors and how that adaptation influences the fidelity of DNA replication. The studies really all fit the rubric that I will tell you about now. Generally, the organism was a specific gene was introduced into the organism. That gene often resulted in some sort of selective advantage. You can think of this as like an antibiotic resistance gene. That gene was transcribed and translated, and if it was made, then it provided resistance to this specific stimulus. In this case, it might be plating on an antibiotic. So in this case, the bacteria survive. So the question was, what happens when this gene is mutated in a way that is difficult to fix? In particular, when single nucleotides are either inserted or deleted. In this case, of course, the gene, once it gets to that point of the insertion, it eventually hits a premature stop codon, so you get a fraction of the gene. This no longer results in any sort of resistance, so the bacterium dies. But because the selection pressure is so strong, with some frequency, you get spontaneous mutations that result in fixing this issue and survival on the stressor, in this case, again, an antibiotic. So the expectation here might be that if the researchers go and sequence this gene, they'll find that the gene has been corrected to the appropriate sequence. But what they were surprised to see is that in the vast majority of instances, that isn't what happened. When they then sequenced the plasmids were perfectly fine. They still had the +1 insertion that they introduced. But then when they tried to track down where the responsible mutation that resulted in this resistance came from, they found that it almost always localized to tRNAs that had been mutated to acquire an additional base in their anticodon loop. And what they observed from this is by comparing the sequences of the insertion to the sequences that had been evolved in the tRNA, that there seemed to be a pretty close correspondence between those sequences, suggesting that these tRNAs are now able to read that 4-base sequence. And so you can see here, this is just a summary of some of the studies that showcase this, with red being the position that was mutated in some of these organisms. And so historically, this was sort of observed as an interesting ability of tRNAs to accommodate a 4-base decoding event. But from the perspective of genetic code expansion, it's been this really interesting approach to potentially supplement the genetic code with new information that operates in this quadruplet codon frame. Unfortunately, what these researchers and many, many others have observed later on is this process is incredibly inefficient. And I'm now summarizing a lot of data to tell you a little bit about how many people think about quadruplet decoding and highlight why this process is inefficient. So imagine a ribosome. Ribosomes have 3 canonical sites, the A, P, and E site. The A is where the tRNA is selected. The P is where the peptidyl bond is being formed, and the E site is the exit channel of the ribosome. So the ribosome comes along and it finds this quadruplet codon. It now has a choice to make. Most of the time, it's going to make the choice where it takes a canonical triplet tRNA, puts it in the A site, and of course, because we've introduced this quadruplet codon in the gene of interest, everything downstream is still triplets, and as a result, this would result in a mistranslation event, an incorrect selection of the frame that we want to maintain. So this results in a truncated protein and through eventually hitting a premature stop codon. If what we want happens, then this quadruplet decoding tRNA, or what we call qtRNA, comes in. It hybridizes with the quadruplet codon in the A site. It doesn't do a particularly good job at this. There sort of multiple models for what's happening at this stage, but suffice it to say that not all 4 bases are fully interacting. The ribosome can then sort of survey this and choose to throw it back out of the ribosome and try to find an appropriate complementary tRNA. But if it does proceed, the tRNA moves to the P site, the ribosome moves on the mRNA, a new bond potentially forms. The interaction with that 4th base may or may not be sort of optimized at this point. But if the ribosome does find a way to fit it, then translation proceeds normally and the frame is restored after this quadruplet codon. If that doesn't happen, then again, a triplet tRNA comes in, it competes with this process and results in incorrect frame maintenance and a truncated protein. But if it does happen, then a triplet tRNA comes in and translation resumes after this stage. So you can see hopefully from this that there's a lot of possibilities for the ribosome to abandon this process, which results in the overall poor efficiency, or of course for the truncated proteins that you see. And so we got interested in trying to define what are the blue arrows that we could tune to increase the overall efficiency of this process. So we broke down the problem into a number of steps, how the decoding happens, potential competition issues, what are the local sequence requirements that influence this process, and how can we leverage the abundance of pre-existing tRNAs to our advantage to overcome some of these limitations. So in our first study, we built very simple circuits to explore this question. We built plasmids that look like the ones that you see on the left. On the top is a circuit that makes the quadruplet decoding tRNA that we've engineered, and on the bottom is a given reporter gene that can be anything that you want it to be. I'm just showing you examples here of it being luciferase or GFP because those are relatively easy to monitor. And when we do this, by selecting certain tRNA scaffolds and just mutating their anticodon loops, we make a really interesting observation that actually aligns quite well with prior work, which is that much of the time this approach doesn't work. So I'll just walk you through how to interpret this. These are all tRNAs that, it starts with a single tRNA that puts in tyrosine. We've engineered now its anticodon loop to be complementary to the sequences that you see at the bottom. And so what I hope you can see is that in this case, only this codon works, most of these others don't, but perhaps most importantly, the y-axis is normalized to natural triplet protein translation. So overall, the efficiency is quite poor. So the result is bad, it's very unpredictable. You can sort of flip this paradigm to use the same codon, but different tRNA scaffolds, and more or less the same answer arises, which is that in a significant fraction of the instances it doesn't work, and in those that it does, is quite poor overall efficiency. So this was sort of the state of the art, that serendipitously discovered tRNAs do work. In general, it's quite difficult to engineer them, but potentially we could define rules that allow us to evolve or engineer these tRNAs to have higher efficiencies. So the way that we did this is using a technology that I helped co-develop called PACE, or phage-assisted continuous evolution. I'll quickly run through this because it's relatively old at this point. Fundamentally, what PACE allows you to do is to couple the function of an evolving biomolecule, in our case that might be a quadruplet decoding tRNA, to the production of an important protein for the viral life cycle, which is called P3. So somehow an evolving gene of interest, which is included on the phage genome, gets transcribed and translated potentially. In our case, it would just be transcription. It has to somehow affect a circuit that results in production of P3. And if we couple these things well, then every time our evolving biomolecule acquires a beneficial mutation, that phage propagates better. We also overexpress a number of mutagenic proteins to increase the mutation rate in the system. This is what allows the evolving biomolecule to actually evolve. And then we set up the system so that it sort of functions autonomously. We initiate the experiment by adding some phage. That phage genome is now inside the cells. Mutations are acquired all over the place. If the tRNA is functional, then it results in production of more viral progeny. If it's not functional, then that key protein is not made, and any virus that's produced is actually not infectious. And then we just automate everything in this cycle such that increasingly functional biomolecules are retained under this continuously diluting vessel that we call the lagoon. And this allows us to do directed evolution very, very quickly. So compared to traditional strategies where you would need a few days to a week to do a single round of directed evolution, PACE allows us to do about 50 or more potentially rounds of directed evolution every 24 hours. And because this is done in such a way where the flow rate is tuned to allow the phage to live in the lagoon, but on average, no single bacterium to live for that long in the lagoon. Mutations only really accumulate in the phage genome. Okay, so applied to our quadruplet decoding tRNAs, it looks something like this. The phage genome houses that tRNA, it reads a particular quadruplet codon, and we engineer that key protein, that key gene, gene 3, to have a quadruplet codon in it. And when it translates this, it makes that missing protein, it results in viral progeny, and those go in that cycle. Now, because the tRNA is very small, it's only about 70 or so bases, we can do these experiments in only about a day of pace, and they acquire a number of mutations that allows them to dramatically improve their decoding efficiency. So in each case, this is a single tRNA, the gray is the starting point, the red is the evolved variant, and the y-axis is still the same. So this is now compared again to triplet codon translation. And what I hope you can see is that we get dramatic improvements in overall decoding efficiency, and in many cases, those are actually now comparable to triplet decoding. When we look at what actually happened to these tRNAs, some mutations we can reconcile. So for example, in this particular variant, you can potentially anticipate that the red mutations happening so close to the quadruplet codon makes sense to somehow improve quadruplet decoding. But in other cases, the mutations are quite distant, and it can be quite difficult to rationalize exactly how these improvements are being made. Nonetheless, directed evolution gives us this venue where we can improve these activities in an unbiased manner and achieve some of the results that we wanted. Okay, so another really interesting attribute of these tRNAs is that they're mutually orthogonal, meaning if we have multiple tRNAs and multiple corresponding quadruplet codons, do any of them crosstalk? Unfortunately, what we find is that when we have a tRNA that reads a certain codon, it only reads its own codon, but none of these other ones. And hopefully you can see this diagonal is the only thing that's functional here. All of the bottom 5 read quadruplet codons that are UAGA. And so this is the only codon that they can read. In addition to that, our tRNAs have evolved a really exciting attribute, which is something we call processivity. Meaning, if you have multiple different quadruplet codons next to one another, can those be translated by a ribosome inside of live cells? And fortunately, our pace-evolved tRNAs can do this quite productively, translating up to 5 or sometimes even 6 quadruplet codons back to back. And one thing I just want to highlight here is that fundamentally what the tRNA is doing is it's forcing the ribosome to remain in that frame processively. And it tells us something very interesting, which is that the ribosome is actually an adept catalyst at reading quadruplet codons, not necessarily just triplet codons. Okay, so going back to our initial goals, we wanted to do multiplexing to incorporate potentially interesting building blocks and like non-canonical amino acids. So the first question we wanted to ask here is, can we multiplex these? We have all of these parts. They seem to work pretty well. They don't crosstalk. And you can see some of the results here. When we do a single incorporation event, the efficiencies are overall quite high. But as we start to stack these, the efficiency plummets, getting very, very close to our limit of detection even with this assay. And this is perhaps not surprising because what we're really asking the ribosome to do is it's reading a triplet code until it hits our 4-base codon, and then it reads that, and then it resumes a triplet code again after that point. And so statistically, if there was a failure at each one of those steps, overall the efficiency should be a multiplier of all of those inefficiencies. And that's exactly what we see here. But the fact that we can even get this to work at all is pretty exciting. And it suggests that there might be new opportunities for us to improve this even further. So at this point, we took a step back and tried to understand exactly what sort of knobs people had manipulated in translation inside of living cells to try to increase quadruplet decoding more predictably and more efficiently. And I'll just summarize decades of work to say that virtually every attribute of translation had been explored with a single exception, and that's the contribution of mRNA to quadruplet decoding. Now, there's a caveat here, because imagine that you want to incorporate a given non-canonical building block. And if the element that you need to modify to allow that incorporation to happen better is at the mRNA, then you potentially risk changing the overall sequence of the protein that you're trying to make. So to explore this, we recognized this issue and did this experiment in such a way that only provided access to solutions that would not change the underlying protein sequence. And that's by exploring how codon usage influences overall quadruplet decoding. The experiment was very simple. We just took GFP, we chose a position that we know is tolerant to non-canonical amino acids. We either put in a quadruplet codon or used an amber stop codon as a triplet control. And then we built a library where each one of the positions, 5 amino acids upstream or downstream of that amino acid, was mutated to all possible codons for that given amino acid. Now, this library is relatively small. It's only on the order of about 1 million members. And we transformed this into our genetic code expansion strain and then just did facts. So everything that is green is something that has read this codon, triplet or quadruplet. We bin those into things that are really fluorescent or poorly fluorescent. And then we compare that with the unsorted library by NGS and ask what elements, what codon usage modifications allowed this process to become more efficient. So I'll show you the amber data first. So this is a non-canonical amino acid that is a variant of lysine. It's incorporated by this synthetase, and the tRNA reads this amber stop codon. We're exclusively, in this case, looking at the +1 position, so the position immediately downstream of the quadruplet codon. Happens to be an isoleucine, which has 3 codons, AUA, AUC, and AUU. The library has about a third distribution of each one, and no matter what bin we look at, there is no difference between any of those at that position. So this means that whatever thing was influencing the fluorescence that made it more efficiency for amber, it actually had nothing to do with the codon usage at that position. But the answer is dramatically different when we look at quadruplet decoding. So for canonical amino acids or non-canonical amino acids that read the same or different quadruplet codons that you can see here, the distribution changes depending on what bin we look at. So the libraries, again, pretty nicely covered, about a third in each case. When we look at the bottom bins, these blue groups are reduced and they're increased in the top bins, and this happens to be the case in all 3 of these settings. And what I didn't tell you is that the blue bins are the high usage isoleucine codon, and the gray bin is the low usage isoleucine codon. And so over a number of sort of subsequent experiments, what we figured out is that a high usage codon immediately downstream of that quadruplet decoding event dramatically increases overall decoding efficiency. And you can reconcile this a little bit, although we don't have data to support it, by the model that I told you about a few slides ago, where the ribosome can sort of choose to assess if this is a good fit or not. And our hypothesis is once the quadruplet decoding tRNA enters the A site of the ribosome and it's sort of going back and forth trying to create that new bond, if the next tRNA comes in very quickly, then it sort of forces the ribosome to commit to that process, resulting in an apparent overall increase. So we made a number of other observations in this. Another finding that we made is that sometimes there's crosstalk depending on the quadruplet codon that you've chosen, because every quadruplet codon has a triplet codon in the first 3 positions. And so we've developed a number of strategies now to address all of these problems, namely that we constrain the genetic code that we're working in. So we do 2 things. The first is we define a single triplet codon for each of the 20 canonical amino acids that gets rid of any sort of crosstalk issues that we might have. And the other thing that we do is each of those is exclusively the highest usage codon for that canonical amino acid, such that in any gene of interest, we can change a triplet to a quadruplet and capitalize on that increase in overall translation efficiency. So we built circuits that look like this. Everything is being controlled with inducers to give us the maximum sort of ability to tune the system. tRNAs and the synthetases are made, they take the non-canonical, they translate a given reporter. And this is just to convince you that in traditional genetic code expansion, we get a system that looks like this, where the output is dependent on all 3 things happening, the tRNA, the synthetase, and the amino acid being added. In our recoded framework, which only uses the amino acids that you see up top here, we see that it's basically the same result, and that the cell is happy to translate using that compressed genetic code. This turns out to be quite enabling. We can apply this to virtually any tRNA synthetase pair. This is just showing you that these systems are all compatible with that recoded framework. In this case, they're all just reading AMBER so that we can look at the crosstalk between different tRNAs and different synthetases. We use this approach to nominate our favorite tRNA synthetase pairs. You can see this in this box. These all incorporate non-canonical amino acids, and you can see that the efficiency is really high on the diagonal. There's basically no crosstalk for these top 5, which is what I'll be telling you about moving forward. and that we can also show that they're mutually orthogonal with the different non-canonical amino acids that we add to the system. So there's sort of three-way mutually orthogonal at the tRNA level, at the synthetase level, and at the non-canonical amino acid level. Okay, so we have all our tools now. Can we go back to the original goal, which is building these new to nature molecules that allow us to expand the genetic code up to many, many different non-canonical amino acids? So to do that, we basically optimized each one of these sets. In particular, we recognize that the tRNA is a major determinant of how the ribosome reads the genetic code. So we actually just focused all our evolutionary pressure on the tRNAs. We took those 5 tRNA synthetase sets. They're color-coded here, and these colors will be the same for all the slides I show you from now on. Each one has a defined synthetase, defined tRNA, and defined codons. Wherever you see that there are multiple, we actually just tried different permutations of that design to see which would work best. And in some cases, we had some insight from prior work that told us that certain combinations would be the most preferred. So we randomized the tRNAs, we subjected them to evolution by chlor selections, we sequenced things that came out, we validated them, and we tried to understand exactly how those sequences influence the function. So this is a summary of a lot of work. These are all the different evolution campaigns that we applied to these 5 tRNA synthetase pairs. The way to read each of these graphs is at the bottom, it's plus or minus the non-canonical amino acid for that pair. Color code is still the same as I showed you on the previous slide. The structures are shown in each case, and the colored bases are the ones that we evolved over the course of this study. The gray is the starting point, and the colored is the evolved final product. So there's a couple of takeaways that I want to highlight. The first is that in many cases, this improvement is quite dramatic. The second is that the dynamic range is quite large. This is pretty excellent in terms of selectivity for the given non-canonical amino acid that we care about. And perhaps most importantly, the y-axis is still natural triplet protein translation. And so what this means is that now we're able to achieve values that rival natural triplet protein translation. But remember now, the ribosome is reading a 4-base sequence, and it's incorporating a non-canonical amino acid in each of these cases. The other really cool thing about this is that each one of these reads a unique quadruplet codon. You can see the labeling here on the y-axis. And what that means is we could potentially start to multiplex some of these incorporation events. So to explore that, we first wanted to understand what is the amino acid scope that our system provides access to. And so in this experiment, the x-axis is the different non-canonical amino acids that we tested. All of the side chains are shown on the right, so you can see it's a pretty broad repertoire. Each one of the graphs is a different tRNA synthetase pair that reads its cognate quadruplet codon. And so one takeaway I want you to get from this is that each of our tRNA synthetase pairs has a sort of defined but not single amino acid scope. And that's really useful because what it tells us is that downstream, we can start to define DNA sequences that generate multiple unique products depending on what non-canonicals we add to the mix. They often all have sort of structural similarity that makes sense. So the easiest one is the one down here. This is a tryptophan synthetase that has been engineered. It reads the quadruplet codon CGGA, and it only puts in these 4 building blocks. When we look at the numbers over here, you see that they all look like tryptophan analogs because the active site has only been slightly modified to accommodate related structures. Okay, so that was sort of a long-winded segment to show you how we built out all of these parts. But now we have synthetases that charge a number of exciting building blocks, tRNAs that read quadruplet codons really effectively, and each of them is corresponding to a unique quadruplet codon. So when we put all of this stuff together and take it back to macrocycles, we can capitalize on one of the systems that I told you about before, which is called Cyclops, that uses a split intine strategy to circularize peptides, and in doing so, create macrocycles with high efficiency inside of live cells. In our context, what we can do now is assign each of these to a quadruplet codon, and because our systems are relatively processive, the ribosome will remain in this frame as long as the tRNA and the amino acid and the synthetase are tuned to have the right specificity and activity. It will create macrocycles that look like this, where each one of the side chains is one that we have defined that is new to nature. So just to show you how the system works, We build circuits that allow us to control the expression of these split intine systems. We make tRNAs and the synthetases. This is just a model canonical amino acid only macrocycle. It has this sequence CLLFEY. All we do is take the cell pellet, resuspend it in a semi-acetonitrile, lyse the cells, shoot that on the LCMS, and we see these really large peaks suggesting that the overall efficiency of the system is quite high. If we then mutate that split intine, we see nothing on the LCMS. And so this is a nice sort of negative control to show us that we're able to make the macrocycles that we want. We can layer this now with our genetic code expansion resources. So we make all the tRNAs and all the synthetases. The intine does its work and it makes these macrocyclic small molecules. So I'll just show you some controls before we get to the really exciting part. So each of these is a CLLFEY analog where a single one of the codons has been swapped to a quadruplet codon. The colors are still the same. Each one is a different tRNA synthetase pair. All of these are the same LC traces. The top is with the non-canonical amino acid. The middle is without. And the bottom one is where the intine has been mutated, so it can't cyclize anymore. And so hopefully you can see that there's a peak on top, and then the other two boxes don't have anything in them. But because the systems are relatively efficient, we can start to do more complicated things. So we can introduce the same amino acid multiple times in a macrocycle, or we can even start to multiplex these to make increasingly unnatural products with high yield and high efficiency. Overall, the system makes a pretty decent concentration of these molecules. So the way that we figured this out is for some of the macrocycles that I'm showing you here, we made solid phase peptide synthesis standards. We compared them to the things that we're seeing from the cells. So up top is the standard. Here is the product from E. coli cells, and you can see that the peak is the same in all cases. And by creating a standard curve from this solid phase peptide synthesis product, we can actually find that we're making on the order of 1 to 10 micrograms from only 0.5 ml E. coli culture. And that's extremely high, especially in the natural product space. And importantly, when we compare this with the lead time for solid phase peptide synthesis from perhaps one of the best vendors in the world, it's on the order of many weeks, whereas we can do this experiment in a couple of days, and all we need is to order a new piece of DNA for the macrocycle. More recently, in unpublished work, we've been able to push this system even further. We've engineered the tRNAs even more, the synthetases to have even higher function, and we can now access even more unnatural looking small molecules like these. So in this case, the tRNA is allowing us to incorporate multiple of the same non-canonical amino acid back to back. This cysteine is required for macrocycle formation. That's why it's the only canonical that's left here. But everything else has been modified, and hopefully you can see we're making these really interesting molecules. But also we can start to make some more sort of defined polymers like these AB type polymers, or where each one of these positions has been modified to be a unique quadruplet codon that incorporates its unique non-canonical amino acid. And in other work that I don't have time to show you today, we've also been able to show that the scope of the amino acids that I highlighted is compatible with this. So the same DNA sequence can allow us to make many different macrocycles depending on the different non-canonical amino acids that we add and the placement within that macrocycle sequence. So with that, I'll just sort of summarize back on this model of how we think quadruplet decoding happens. We've done a number of things so far to try to push this. We've developed paste that is compatible with qtRNAs to make them better substrates for the ribosome. We've compressed the codons such that we don't have these competition issues as much anymore, and we've eliminated the low usage codons that allow us to circumvent potentially off-target decoding events. And in unpublished work, we've also been able to show that by balancing the expression of all of these things, we can unlock even higher levels of multiplexability. So where is this going? This is a long-term effort that we've been focusing on now for a couple of years, and we're only a fraction of the way through to where we want to go. In other work, we've been trying to understand exactly what it is that makes a good quadruplet decoding tRNA, and extending this to many, many different tRNAs and learning the rules more broadly, both for the canonical and the non-canonical amino acid spaces. And we've been trying to layer all of these technologies on top of one another such that we can co-evolve all of the necessary machinery, the ribosome, the tRNAs, the synthetases, the various elongation factors, to allow us to now access truly comprehensively modified polymers that all rely on quadruplet decoding with high efficiency inside of live cells. So with that, I'll just summarize by saying that really simple experiments, especially for convoluted systems like genetic code expansion, can actually teach us a lot about how protein translation happens and some of the guiding principles for how to improve some of these processes. We can evolve these tRNAs to make them much more efficient at quadruplet decoding, and we can layer these technologies with others to provide access to fully diversified new to nature molecules with high efficiency and in high yield. Like I said, we're working on a number of different extensions of this work. One of them is trying to understand how we can subject these macrocycles to directed evolution so that we can nominate sequences that have interesting new to nature function, not just sequences and structures. We've also been incorporating new to nature backbones so that the macrocycles that we're making are not just modified on their side chains, but also have really interesting chemistries that might constrain their topologies in interesting ways. So with that, I'll just thank everyone for coming to the presentation. I'll thank Kate again for the really kind invitation. I just want to highlight that everything that I showed you today has been led by two exceptional researchers, Alan Costello, who led the first study, and Cindy Chen, who's sort of taking this work into incorporating higher order multiplexed macrocycles. This is a nice drone video that Scripps paid for to fly through my lab, so I sort of reap the dividends on that, but hopefully you can see, and one of the few times where our lab is so clean, but hopefully it gives you an impression of what the research looks like. I'd like to thank all our funding agencies that have been extremely generous in supporting our work. You for your attention, and I'd be happy to take any questions.
Thank you. I really love the drone video, and I hear you. On videos you can find online of my lab, it looks so clean. That's because we got advanced notice.
That's right.
Awesome. That was a really inspiring talk. This is something that a lot of people in our community have been looking at, this expanding of the chemical diversity. There's quite a bit of questions in chat. Can you see chat?
Yes, let me open that.
If you can read the question, because people watching the recording don't see chat. So
got it. Okay. Do we have ARSs, so synthetases that charge tRNAs in a codon-specific way for quadruplet codons? Yes. So it's an interesting question. Maybe I'll sort of provide some background here first. Genetic code expansion relies on tRNA synthetase pairs that have a very unique attribute, which is that the synthetases themselves do not interact with the codon. This allows the researcher to change the codon at will without having any sort of downstream deficiencies in synthetase amino acylation. The vast majority of tRNAs are not amino acylated in this way. In fact, in E. coli, 17 out of the 20 synthetases physically interact with the quadruplet codon. And as a result, many of those have not been engineered for, or related synthetases have not been engineered for quadruplet decoding, because you would anticipate that the solution space for possible codons is much smaller, because it also has to satisfy what the synthetase wants to interact with. So all of that is to say, there are rare examples where tRNA synthetase pairs are engineered to amino acylate tRNAs in a codon-specific manner. They do exist, but they are rare. Okay, if you have, this is the next question. If you have a quadruplet codon on mRNA, how do you stop canonical tRNA that matches part of the quadruplet codon, incorporating canonical amino acid and messing up the reading frame? That's a great question. So fundamentally, both of these things can happen at the same time. So for a given sequence, if it's a 4-base sequence, a triplet tRNA can come in or a quadruplet tRNA can come in. What makes our system work better is two things. The first is that, and this is something I didn't discuss, is that quadruplet decoding tRNA has to have the first 3 bases correspond to a low usage tRNA. And so what this means is that by overexpressing your tRNA, you outcompete that low usage tRNA. So the system prefers to use your tRNA. The second thing is the one that I talked about, which is that we have that +1 high usage codon that increases the preference for decoding of a quadruplet codon. You might ask, though, if we're doing this in such a way to outcompete the triplet tRNA, how does this not result in any sort of toxicity in the organism? Because that tRNA is being used to translate genes in that organism. And the short answer is we don't know, but we think it doesn't result in any sort of toxicity because low usage codons are often less abundant in highly essential genes. And so even though we may be mistranslating host proteins, in general, those are not essential for viability, so we don't see any deficiencies there. Could paste be used to evolve the ribosome to get better decoding of consecutive quadruple codes? Yes. So we actually had another study a few years ago where we developed what we call oribopase, which allows us to evolve ribosomes, in that case, to have increasing translation kinetics. And as a byproduct of that, we actually found that they get better at incorporating non-canonical amino acids. We haven't applied this to quadruplet codons, but you might anticipate that it would be a pretty easy step to take in that case. Do quadruplet decoding depend on post-translational modifications as much as triplets for fidelity? Or do you have more freedom to use non-modified tRNA given longer recognition sequences? That's a great question. We have no idea, but what I can tell, maybe let me rephrase this question first. tRNAs in most organisms are heavily modified. Pound for pound, they are more modified than any other RNA that exists in a cell. And as a result, when we start to engineer these tRNAs, an interesting question arises, which is, are our evolved tRNAs also modified, or are they immune to that modification? There are some studies that explore this. They don't always agree, but there is evidence that even orthogonal tRNAs from archaea brought into E. coli do result in some post-transcriptional modifications. How that is influenced by evolution in that host, we don't know, but we're starting to initiate collaborations that will allow us to explore this in much higher detail and also higher throughput. So stay tuned for that. Okay, let's see what else. Can you comment on those evolved qtRNAs that have single insertions or deletions in the stem? I don't think I've ever seen tRNAs that look like that, and it's funny that those seem to work better. That's a great observation. So we don't know where those come from or even what they do, but what I can tell you is the tRNAs start by having perfectly complementary stems. We then order oligos to randomize those stems and then subject them to evolution. And what we find in more than one case is that those stem acquire indels that are important for function. The only way those can arise is either from errors in the DNA synthesis or just random indels that happen in E. coli as the plasmid is being propagated. Why it happens is a little weird to us. The fact that we see it more than once suggests that there may be some interesting biology there, but outside of that, it's really difficult for me to speculate on exactly why that's the case. But we have confirmed in subsequent studies, if we introduce a complementary base or if we remove that sort of bulge that exists in the stem, that both of those result in ablated function. So they are clearly important for activity. Could in future quadruplet codons infused into plasmids as separated retranslational material locked in collagen capsule with aptamers for bio? Okay, I'm going to rephrase this question. Basically, can tRNAs serve as therapeutics if they were encapsulated and delivered into cell? I think the answer is definitely yes. There's already a lot of work that's being done on creating different delivery vehicles for engineered tRNAs that are used to decode missense mutations in human cells and probably in downstream sort of clinical applications. I think that's an active area of research. There are many companies that are also exploring this. So it would be difficult to imagine that the answer to your question is no. So I think it's definitely something that's happening in the works. We're not actively working in that space. Do you believe that we may have other planets where quadruplet codons are the main players and triplet is exception? That one's from Taesak. I don't know. It's hard. I think that the most definitive answer that you can give, that anyone can give about anything in biology, is that it's good enough. So why are there 64 codons? Why are they triplets? Why are there 20 amino acids? It just works. And biology had access to those parts and it evolved to use them. That doesn't necessarily mean that quadruplet codons wouldn't be a viable means of conveying genetic information on this planet or somewhere else. So I don't know what the answer is, but I think it's definitely conceivable. Why it didn't happen in that way is a much more difficult philosophical question to answer.
That's the perfect answer to me for now. But the reason I'm asked that question is, 64 is sufficient enough, or probably probability, perspective, probably, I don't know. I mean, probably the good or optimum, or I don't know, at least local optimal or global optimal, I don't know. If we have the four, quadruple, codons, we may have a more diversity. I don't know,
Yeah.
or less
Yeah.
diversity.
So I'll just sort of say this. There's a number of examples now, historically pioneered by Pete Schultz, but now more recently, over the past many years, really elaborated on by Anthony Green, showing that if you judiciously pick the right non-canonical amino acid, you can access function that exists beyond nature. So from that perspective alone, you might argue that expanding the genetic code, letting biology have many different non-canonical building blocks would have a fitness advantage eventually. It's hard to predict what those examples are. The only, and I should sort of say that his examples are, he knows the amino acid that would improve the function or have the function, and he knows where to put it in the gene of interest. As far as I know, there's only a single example, and this was work that was done by Andy Ellington more than 10 years ago now, maybe 12 or 13 years ago, to ask a related question, which is, if you give a system the ability to choose to use a non-canonical amino acid, does it do that? And does that provide a fitness advantage? And what they did is they created these systems where they have a strain, it can do genetic code expansion, they give it the non-canonical amino acid, and the only thing that they do is they propagate a bacteriophage in that strain. There is no selection pressure outside of propagation. And what they find is that in at least one population, that population very quickly converges on a solution that does use that non-canonical amino acid. The resulting phage is more fit than the wild type phage, and the only selection pressure was just propagation. There's no incentive for it to use that amino acid. So again, there's many different pieces of information there, but all of it seems to suggest that if you give a system a large enough repertoire of unnatural building blocks and enough time, inevitably there has to be solutions that provide a fitness advantage that also rely on the non-canonical amino acids. It's just they haven't existed in nature, so the solution space might be quite distant. And there may not be an immediate benefit. But long term, I think the answer is that they would be beneficial. We just have to think of the right instances.
Yeah, that's what I'm thinking when I ask that question. We may have some planet has superheroes. All of them is superheroes, basically.
It's possible, yeah. We have to find that planet first.
Thank you.
Thanks.
That's a good goal. That's a good goal to end on. Let's go find a planet with quadruple codon life.
That's right.
All right. Thanks so much for an amazing talk, and thanks everyone for great discussion.
Absolutely. It's my pleasure.
Have a great rest of your week, and see you next week.
Thank you. Take care, everyone. Bye.